Our next topic is genetic counseling. There are two types of cell division that occur in the human body. The first is mitosis. This is the type of cell division that occurs in your somatic cells, or your body cells. The purpose of mitosis is to allow for growth. So think of an infant growing into a child and an adult. You're going to need more cells for growth to occur. Mitosis also allows for replacements of cells that get old or worn out. Mitosis also allows for repair. So if you were to cut your finger or break a bone, mitosis would generate new cells that would enable tissue repair. The outcome of mitosis is the daughter cells. These are cells that are generated, are identical to the parent cells. All somatic cells in the human body contain 46 chromosomes. 46 is considered to be the diploid number of cells. This slide shows mitosis in a sea urchin. Sea urchin begins as a single cell. Then it divides to form 2 cells, 4 cells, 8 cells, and this can actually be viewed using a scanning electron microscope. Next, a small ball of cells forms. After that, many more rounds of cell division, you have a complex multicellular organism, which is a mature sea urchin. This is similar to the process that occurs in human development. Meiosis is a type of cell division that forms gametes. Gametes are sperm and egg cells. This is a type of reduction division because the cells that are generated are haploid. Haploid means a chromosome number is halved, and so sperm and egg cells have 23 chromosomes. During this process, there's a shuffling of genes that occurs on homologous chromosomes. Remember, homologous chromosomes are chromosomes that contain the same genes in the same order. However, some of these genes may have alternate forms or alleles. This causes a type of genetic shuffling that produces greater variation in the offspring that are produced. Therefore, when a child is born, they have pairs of homologous chromosomes which may contain alternate forms of each gene or alleles. Both males and females have 23 pairs of chromosomes. The first 22 pairs are called the autosomes, and these chromosomes code for most traits. The last pair of chromosomes, the 23rd pair, are the sex chromosomes. Females are XX and males are designated as XY. The human life cycle alternates between haploid and diploid cells. Meiosis produces haploid sperm and egg cells. When fertilization occurs, the zygote that is formed is diploid. This diploid organism is then going to grow, and then adulthood generate sperm or egg cells, which are again haploid. This chart summarizes the life cycle of humans. So as we mentioned, mitosis is a type of cell division that allows for growth. So here we have our 2 adult humans, male and a female, and meiosis occurs in the testes and ovaries. This reduces the chromosome number to 23, and we have an egg that has 23 chromosomes, a sperm that has 23 chromosomes, and then fertilization generates a zygote, which has 46 chromosomes. The zygote is then going to undergo mitosis and then generate another adult human. This diagram shows a karyotype. A karyotype is an arrangement of chromosomes in order. As I mentioned before, that the autosomes are the first 22 pairs of chromosomes, and the 23rd pair are the sex chromosomes. Here we can see that this individual is a female. There are 2 X chromosomes present. Also notice that the chromosomes, the autosomes, are arranged by size. The first pair being the largest, down to the 2nd, 3rd, 4th, down to the 22nd, which is the smallest pair of chromosomes. You can see that the dye causes staining in the banding pattern, and that's makes it easier to align the chromosomes together to match one homologous chromosome to the other. Here's another karyotype, and this shows a male. You can see because of the presence of the X and Y chromosome. Sometimes the chromosomes are also grouped by letters into sets. And again, you can see the chromosomes are paired, the largest pair down to the smallest. A karyotype can be used to do genetic testing. Genetic testing can be done on fetal cells, can be done on children and adults. One type of abnormal chromosome inheritance is non-disjunction. When meiosis occurs, the chromosomes initially replicate, and then they divide, and then divide again to reach the haploid number. This is called the process of meiosis 1 and meiosis 2. So there's really two locations where the chromosomes cannot separate properly. And this is going to then cause a gamete, a sperm or an egg, to have 2 of a chromosome instead of 1. When fertilization occurs, the resulting zygote can have a trisomy, which means it has 3 copies of a chromosome, or it can be a monosomy, which means one of the sperm and egg cells lack that chromosome. So the fertilized egg now only has 1 copy. This chart is a little complicated, and we really do not go into meiosis in great detail. That's something you would do more in a general biology class. But what we're interested in just understanding is the outcome is that you can either end up with a sperm or an egg cell that has an extra chromosome or is short a chromosome. So you have, say, N+1 or N-1. Down syndrome is the most common trisomy. It's called trisomy 21, which means there are 3 of the 21st chromosome. A syndrome means symptoms that run together. So for any of the syndromes that we're going to discuss, there's usually a series of characteristics. And not each individual will show each characteristic in the same way. So the recognizable characteristics of Down syndrome is short stature, eye folds, stubby fingers, mental disabilities. There's also potential heart or respiratory problems. But again, saying that it's a syndrome, there are individuals with Down syndrome that have an average or above average level of intelligence. The chance of a woman having a Down syndrome child increases rapidly, starting at age 40. In females, the oocytes develop prior to birth, and they actually start to undergo the process of meiosis before the female is born. At puberty, several of the eggs will start to complete the process. One egg becomes a dominant, and then ovulation occurs. As a female ages, the chromosomes become more sticky, and that causes non-disjunction more likely to occur. So think of the difference between a female that has a baby of age 20 and a female that has a baby at age 40. Again, all the oocytes have been already present, so you have eggs that are developing that are 20 years old versus eggs that are 40 years old. So as the eggs become older, there's more likely for chromosome anomalies to occur as meiosis is completed. Let's look at some abnormalities in the number of sex chromosomes. This is where an individual has too many or not enough X or Y chromosomes. Remember, it's the Y chromosome that determines maleness. This is due to the presence of the S or Y gene on the Y chromosome. During embryonic development, this gene turns on the production of testosterone, which causes the production of primary male sex characteristics. Therefore, an individual that has one Y chromosome is automatically genetically a male. Turner syndrome is a female that only has one X chromosome. Let's look at the designation that is used in describing the karyotype. 45 means that this individual has only 45 chromosomes instead of the normal number of 46. Then a comma is in use, and then you designate exactly what the abnormality is. So the XO, the O means the absence of another X chromosome. Again, if an individual has a Y chromosome, they're a male. They don't have a Y chromosome, they're a female. So genetically, this individual is characterized as being female. Klinefelter syndrome is a male that has an extra X chromosome. So the designation 47XXY is used. Let's look at some of the characteristics of an individual with Turner syndrome and Kleiner syndrome. The female with Turner syndrome has a web-like neck, less developed breasts, and less developed ovaries. The male with Klinefelter syndrome has usually very little facial hair, may have some breast development, very long arms, less developed testes, and very long legs. Individuals with Turner and Klinefelter syndrome are usually sterile. XYY syndrome is very interesting. It previously used to be called the supermale syndrome, and when I had genetics, I say the dark ages of biology in the 1980s, we actually referred to it as the criminal male syndrome. Again, remember, syndrome means symptoms that run together. Not every individual that has the XYY syndrome is going to show all of these characteristics. Okay? These individuals, males, are very tall in stature, very muscular, have learning difficulties, and tend to be somewhat more aggressive. So the reason why it was called criminal male syndrome is if you're very big, very aggressive, and not very smart, you do have a tendency to perhaps do things that would cause you to be incarcerated. It's more politically correct now just to call it the XYY syndrome. Genetic counseling can be done to help couples understand the mode of inheritance. This typically occurs when there's a genetic abnormality that's found in one or both of the families. Ultrasound can be done to detect a variety of serious anatomical abnormalities, such as neural defects, a cleft lip or palate. It can indicate some types of congenital heart defects, congenital tumors, gastrointestinal or kidney malformations, or limb defects. Fetal cells can be extracted by amniocentesis or chorionic villus sampling. Some fetal cells can also be collected from the mother's blood. Amniocentesis is a process where amniotic fluid is extracted between the 14th and 17th week of pregnancy. Amniotic fluid protects the developing fetus by cushioning blows to the mother's abdomen. It also allows for ease of fetal movement and promotes skeletal and muscular development. There are only a few disorders that exist due to change in chromosomal number. More variants are possible, but do not give rise to viable offspring. More common are disorders that are due to changes in individual chromosomal structure. Changes in chromosome structure include breakage, deletions, duplications, translocations, and inversions. A deletion occurs when a chromosome loses its end, or there is two breaks that result in the loss of an internal segment. Williams syndrome is due to a loss of the end of the 17th chromosome. This causes cardiovascular disease, developmental delays, increased verbal ability, and affinity for music. Houda-Koss syndrome is where the chromosome number 5, a small section of the end, is lost. The name comes from the term cry of the cat due to the sound that's made when these infants cry. They're born with a small head, very low muscle tone, low set ears, small jaws, and a round face. (22q13) is the name of this disorder, and it refers to the location of deletion. It's a microdeletion on chromosome number 12, the 13th region. This disorder, however, occurs spontaneously. It's not inherited. A duplication is where a chromosomal segment is repeated. The individual therefore has more than two alleles for a certain trait. The IMP-dup15 is an inverted duplication on the 15th chromosome. This specific region, called 15q11q13, is known for its instability. It's highly susceptible to rearrangement. Children who have this disorder display low muscle tone, developmental delay, intellectual disabilities, may have epilepsy or be autistic. A translocation is exchange of segments between non-homologous chromosomal pairs. The individual has a normal amount of genetic material and may be totally healthy. About 5% of Down syndrome cases are caused by translocation in a previous generation between chromosomes 21 and 14. This slide shows an example of reciprocal translocation. An inversion is when the segment of a chromosome is turned or does a 180. This can lead to altered gene activity if the inversion disrupts control of gene expression. Many other genetic disorders exist that are not due to chromosomal abnormalities, but defective genes. A disorder due to defective gene indicates that not enough of specific protein is being manufactured, or the protein that's being manufactured is defective. DNA tests are done to look for genetic markers. Genetic markers indicate abnormal alleles. Using in vitro fertilization, embryos can be generated that can be screened for genetic diseases prior to their implantation. During in vitro fertilization, a sperm is used to fertilize an egg. This one-celled zygote then undergoes mitosis. When it is at the 8-celled embryonic stage, one of the cells can be removed. This cell can be analyzed to see if the embryo is genetically healthy. While this procedure is typically done to screen for genetic abnormalities, it can also be done to select for a particular gender. This guarantees a boy or a girl baby. This is most often used in situations of family balancing. This procedure has led to a slippery slope. Our embryos now can be tested for obesity genes, for other non-life-threatening diseases, for height, eye color, hair color. Will this lead to eugenics, the science of improving the population by controlled breeding to increase the occurrence of what is considered to be desired traits? An adult can also have a genetic profile determined. This gives an individual a complete list of all their genotypes and indicates the presence of any mutant genes. An example of this would be the BRCA1 or BRCA2 breast cancer genes. A sample of DNA is also going to be analyzed to determine the risks of developing common diseases, such as heart disease, Parkinson's disease, or diabetes. There are some possible misuses of this information. First of all, if an individual were to find out that they had a very low risk of contracting heart disease, would that make them stop exercising and alter their diet? Another misuse is what if this information fell into the hands of your employer? Or, worse yet, if you were applying for a job and they found out that, for instance, you were diabetic and that you were at increased risk of having a heart attack, would that prevent you from potentially getting employment? Also, if you applied for a life insurance policy or just regular health insurance, if the insurers got hold of your genetic profile, it may affect your rates or even your abilities to get these types of insurance. 23andMe is a company. With a simple swab of your cheek epithelial cells, you can find a little bit more about your ancestry, your nationality, and also find out if you're carrying any mutant genes.